So where we left off on Friday was we were talking about the fact that some chemicals have a tendency to accept electrons. They tend to be reduced. Some chemicals have a tendency to lose electrons. So good examples of chemicals that like to, remember, we could quantify what things like to gain and lose electrons using voltage. More positive voltage means a more favorable reaction. So like fluorine. Fluorine is this classic electron hog. All semester, fluorine's been hogging these electrons. And we can see that if fluorine F2 gains electrons, it has this really, really favorable payoff here of positive 2.87 volts, which is considerable. If you think about it, the amount of work necessary to power pretty high-tech electronics like our laptops and cell phones is 3.4 volts. So that's a considerable amount of electrical potential there. Okay. On the other hand, some materials don't want to gain electrons. For example, sodium and lithium, once they've given up their electrons and they become positively charged, they're good. They're obeying the octet rule. They don't want to change their charge. So those have very, very negative potentials. As we can see, lithium's is right down here at the bottom, in the bottom right-hand corner. That comes with a price tag of negative 3.505 volts. What that means is that you'd need to put in a good deal of work. You would need to facilitate that reaction in order to get it to go. And a classic example of us forcing that reaction is, so batteries have a certain life to them. They do this reaction, they do a favorable redox reaction, electrons are transferred, they go through a wire, they power things, but eventually they get to this equilibrium where they've made their products, the reactants are depleted, and at that point, the reaction stops. And that's when we throw out our laptop battery, right? Lol, JK. At that point, we recharge them. So we plug them into an electrical current, and that electrical current allows this negative voltage reaction to occur. So it forces electrons to go into lithium when it ordinarily wouldn't want to do that. So we basically use electrons to force the reverse reaction. And then once we have it charged back up again, we can unplug it and let it do its thing again. So anyway, a little bit about voltage. So now what we're going to do is, we finished with this slide on Friday. We talked about some different potentials, and y'all rock this clicker question here. We looked at reduction potentials and oxidation potentials. And recall that, so reduction is gaining electrons, oxidation is losing electrons. If you have a reduction reaction and you spin it around, a reduction reaction, if you reverse it, becomes an oxidation reaction. And what happens to the sign of the voltage if we flip around a reaction? Can somebody please tell me what happens if we flip around a reaction? What happens to the sign of the voltage? To the chat. Da, 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 da. Good, it flips as well. So if, for example, let's look at aluminum. So aluminum, in order to reduce it to form aluminum solid, it costs 1.66 volts. But if we were to flip this reaction around, so if instead of doing this reaction, we were to do the following reaction. So if instead we wanted to take solid aluminum and we want it to lose its electrons, become aluminum 3+. In order to find the voltage there, all we have to do is change the sign. So this would become positive 1.66 volts. And is very favorable. In fact, aluminum does oxidize very easily. If you look at like an aluminum can, for example, aluminum is incredibly reactive, but it reacts with oxygen in the air around us. It very easily gets oxidized and forms this aluminum oxide. And it turns out aluminum oxide is a very tough material. It's tough to penetrate. So all aluminum cans actually have this protective layer of aluminum oxide that prevents further oxidation of said can. So anywho, So now what we're going to do is we're going to take these potentials and we're going to put them together to create a battery. And I really enjoyed doing this because it's almost like a little bit of an art class at the same time. We're going to be drawing out a little bit of a diagram here. So let's get to it. Okay, building our battery. So what we're going to do is we're going to start by making our compartments. And I'm going to use a not as practical, usually if we were in class, we would be making these out of beakers. Obviously, most batteries don't have glass beakers included in them, but this will get the job done for showing us how a battery generally is set up. So we have our two compartments. And we're going to build our battery with the materials we chose from the previous slide. So when we ended class on Friday, we said, wow, look at gold. Gold loves to be reduced. Look at this positive 1.69 volts, really favorable. We also said lithium loves to be oxidized, positive 3.05 volts, also really favorable. So we're going to use these and exploit them to create a really, really high voltage battery. It's a baller battery because it's got gold in it, but whatever. Maybe not the most practical because it's a very expensive battery. So we're going to have some sort of electrolyte solution in there that has ions as well. Oftentimes in actual batteries, they use like an electrolyte paste. And so our solutions here have these ions floating around of gold, Au plus, and lithium, Li plus. Okay, let's continue. So, what reactions are taking place? We're reducing gold, which gives us 1.69 volts. And we're also oxidizing lithium, which gives us positive 3.05 volts. So the overall potential, the overall voltage of our battery that we're creating here is just going to be the sum of these two. But wait, the plot thickens. So what's basically happening here is the lithium is giving up electrons and giving it to the gold. So as it is, this is what would happen. And this is a very high-tech movie here. Whoosh, whoosh. So electrons are carried through the wire and they plop themselves on the gold side. So as this is written, and just really quickly, some naming conventions here. Electrons always travel in alphabetical order. So electrons always travel from A to C. And what I mean by that is electrons always travel from the anode to the cathode. Okay, so write these in. This will plop in more elegantly in a moment, but just for the sake of naming them right now. Okay, so our electrons are flowing over to the gold. But would electrons, as the electrons are plopping themselves into that solution, it's getting more and more negative because electrons have a negative charge. So inherently, would an electron, a particle of negative charge, want to travel to a negatively charged solution? Do negative charges attract other negative charges? What are our thoughts here? You have a negative charge and a negative charge. Do they attract each other or repel each other? Let's see what we think. Okay, good, they repel each other. So this would create a fundamental problem because electrons are not going to willingly go into a negatively charged solution. So if we're plopping negative charge there, this is going to short circuit within milliseconds. So the idea behind a short circuit is you have this charge differential that prevents electrons from moving, that prevents the flow of charge. So in order to be able to create a battery, you need to do what's called completing the circuit. Basically what you do is you insert something that has cations and anions in it, and it has this selectively permeable membrane that lets the positive charges displace the negative side, and it lets negative charges displace the positive side. So we insert what we call a salt bridge. And the purpose of the salt bridge is to maintain electrical neutrality. Okay, so again, what's basically happening here is we have an anode side. The electrons move over to the cathode, and then our salt bridge keeps things nice and electrically neutral. And just FYI, you don't need to remember this, but in the salt bridge, the anions travel to the anode and the cations travel to the cathode. So basically what we have happening here is, because the electrons are moving their way over into the gold side, the sodium plus then selectively migrates to this side to help create a neutral charge. And on the other side, the nitrate, the NO3 minus, since the electrons are leaving the lithium side, that side would be getting more and more positive. So these can leak into this side. And basically it's just these ions move to create an electrically neutral situation to allow the current to keep going. And then when all is said and done, we can get our potential by adding these together. So our reduction potential is 1.69 volts, our oxidation potential is 3.05 volts, and we end up with a pretty powerful battery, a 4.74 volt battery. So this is our baller battery, our lithium gold battery. For all intents and purposes, it's not super practical, also because solid lithium is explosive if it touches water, but this kind of gets across the general principles. Are there questions about this? About how our battery works and about how we found the voltage of it? Anybody? Okay. So now we're going to talk about something that is related, more or less kind of like a biological battery, so to speak. So let's jump over to, just give me one moment. Again, I have to navigate through 8,000. Okay, there we go. And let's jump to it. All right, so what we're going to be talking about for the rest of class today are concentration cells and neurons. And I'm excited to talk about this because I'm a biologist at heart. Neurons are really, really cool. And a large number of you are communication sciences and disorders majors. So I wanted to make a unit that might at some point, a lot of pathology in communication sciences comes from neuron issues, not being able to transmit electrical signals well. So I'm very excited. I hope that this is pertinent to some of y'all later down the road. I hope that it's interesting to all of y'all. I really enjoy biological applications, and I hope that you do too. So let's get to the general idea of what allows a neuron to function. So we talked about Voltaic cells, where we pair together two different things to create voltage, to create this electric potential, to create a way of pushing electrons from one place to another. Now, it turns out there's also a type of battery, a type of cell called a concentration cell. And concentration cells actually create an electrical signal by having the same two materials paired with each other. So just a little bit of a preface behind this. Here, what we're doing is we have this image of two different nickel compartments. So we have a concentrated nickel solution right here, and we have a dilute nickel concentration here. And we've connected them the same way that we connected our previous voltaic cell. Now, if we look at the oxidation of nickel and the reduction of nickel, what do we know happens if we flip a reaction? If we flip a reaction, the sign of the voltage flips as well. Now, this creates an interesting paradox because if we consider the reduction potential of nickel and the oxidation potential of nickel, they cancel each other out because it's the same magnitude, just opposite signs. So it's negative 0.28 volts plus positive 0.28 volts, which ends up giving us a standard potential of 0. Zero potential means zero electrical signal, means electrons aren't going to be pushed one way or another. So what is it that allows a concentration cell to work? So recall that a standard potential assumes one molarity of everything. In a concentration cell, we break that mode. We don't have one molarity of everything. So it turns out if you pair these things together, electrons will flow until the concentrations on both sides is equal. So we have our dilute side here. We have our dilute solution, and we have our concentrated solution. The electrons will flow until both sides have the same concentration. So we can see over here, 0.5 molar and 0.5 molar. So they're basically evening each other out. And this ends up creating voltage because we're getting the movement of electrons. So we still have an electrical potential here. So if we look at the anode, the side that's losing the electrons, it's the dilute solution. If we look at the cathode, it's the concentrated one. So the cathode will gain electrons, which makes the Ni2 plus concentration go down. The anode will lose electrons, which makes the Ni2 plus concentration go up. And we get this, what looks like a non-reaction, but it turns out it is a reaction because there's two different compartments. Okay. So the key idea here is that by having different concentrations, we end up creating voltage nonetheless. Okay, so now, how does this relate to neurons and cells? And I want to note, I'm going to be going into more detail than you'll be responsible for. I'll highlight the key points, the things that you'll want to be able to do, but to have an understanding of what's happening, we need to kind of go into a little bit of the nitty-gritty behind these things. So cells have, neurons have these pumps where they push sodium onto one side of a cell. And they push sodium onto the outside of a cell. They push potassium onto the inside of a cell. There's all sorts of very specialized pumps. And going back to what we talked about last week, pumps require work in order to function. So we looked at a dam that has water going down it, a waterfall. The water will spontaneously fall down the waterfall, but hence the name waterfall. In order to get water back up to overcome gravity, we would either need to scoop up the water and walk it up if we're being very inefficient about it, or we would have to pump it, which requires energy. Our cells actually also have these pumps that basically, you can think of it as they push the water back up to the top of the waterfall. They push the sodium and the potassium to where they wouldn't ordinarily go. And so we break down a cell. We have our membranes that we've looked at before, and we're looking at two different compartments, just like our voltaic cells had two compartments. There's the outside of the cell, which is called extracellular. And then there's the inside of a cell, which would give the moniker intracellular. And recall that we have in these membranes, we have two different sides of them. We have the lipid core here, and we have the like the phosphate and the polar groups on the outside. So ions will gladly interact with the polar sides, but in our experience, and as you all showed off in the quiz, do ions tend to interact well with hydrocarbons, things like oil? So we know salts dissolve well in water. Do salts dissolve well into oil and hydrocarbons? No, they most assuredly do not. So what this does is it basically creates a shield in between the layer of the cells. So we have this bilayer, and it creates a shield that keeps stuff on one side or another. Now we can open up that shield, as we'll talk about in just a moment. Okay, so we can actually calculate the voltage of our concentration cell that we create through our neuron by using this equation here. And the equation is, so this equation differs. It's based off of something called the Nernst Equation, which is kind of unilateral. You take concentrations, you take charges, you take temperature, and you combine them all into one equation. We're going to look at sort of a simplified version of that will allow us to quickly calculate concentration based off of, or calculate voltage based off of the concentration differences. So at 37 degrees Celsius, which is the general temperature of a mammalian body, we take 0.061 volts and we multiply it by the log base 10 of the inner concentration divided by the outer concentration. Okay, so we take the inside concentration divided by the outside concentration, take the log of that, and then multiply it by 0.061. So I was going to do this part as a clicker question, but I don't know how many of y'all have calculators with you right now. So I'll just walk through the calculation. And you will not need to do this calculation on like a formal assessment. I just want to show you where these numbers come from. So there is our equation. If we look up at our table in the top right-hand corner, so we're looking at sodium here. So sodium has an intracellular concentration of 10 millimolar and an extracellular concentration of 150 millimolar. So we plug those values in, and what we end up getting is the log of anything less than 1 ends up being negative, and when it's all said and done, we get negative 0.070 volts. So because of this concentration, this electrochemical gradient, neurons have this what's called resting potential. They have a baseline voltage of negative 70 volts. And the negative 70 millivolts. And the negative sign is all relative to which side you're looking at. So if we looked at the voltage on the outside, it's actually positive 70 volts. On the inside, it's negative 70 volts. So the negative sign is just kind of relative to what our system is in this case. Are there any questions about this before we move on? Okay, so now let's meet a neuron. Our neurons consist of, so neurons are cells that send electrical signals. And They're pretty complex entities, but we're just going to be looking at three key parts. In this corner, we have these dendrites. It kind of looks like a very wispy plant. And they end up kind of splaying out in all sorts of directions. It kind of looks like a palm tree, actually. Yeah, actually, if we kind of rotate this 100, or if we rotate this 90 degrees, it would look very much like a palm tree. We have our roots on one side, we have our stalk, and then we have, anyway, the palms are called dendrites. Then, the longest part of our neuron is called an axon. And once we get to the end of our axon, we get to this part called the axon terminus. Right? So this is the general anatomy of a neuron. And neurons are cells. Here we have, I'll just point this out. So in here we have the nucleus of our cell where the DNA is held and all that jazz. But we're not going to be focusing on that. So let's talk about the role of each part of the neuron. The dendrites. What do the dendrites do? They receive stimuli. So depending on what type of neuron we're looking at. So some neurons send sensory signals to our brain. Some send motor signals from our brain to the rest of our body. And there's all sorts of different signals that can end up being created. So some neurons specialize in pressure. Like if you feel pressure somewhere, it'll send a signal. Some specialize in heat. Some specialize in smell, in taste, in movement, etc. So depending on what flavor of neuron you're looking at, these will be triggered in different ways. But the dendrites are where the signal goes in. It's the input part. The axon. The axon is basically a highway of sorts for the signal to go down. So the axon can go in some cells, in some neurons, it's about 1 millimeter long. In others, it can be up to 1 meter. So a good example are like the nerves that make our toes move. They go from our spinal column and they run a very, very long distance down the leg to the toes. So these can be either pretty short or very, very long. They first started studying axons in squids and squid tentacles and like anyway, those ones have very, 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 very long axons as well because it goes down their entire tentacle. So these are also sheathed in basically this insulator called myelin. Myelin's important because it helps the signal move more quickly. It creates a more efficient way of sending a signal. A number of neurodegenerative diseases come about because they destroy myelin. And so like multiple sclerosis, for example, is a disease where this myelin sheath, this protective layer that allows signals to go more quickly, ends up being eaten away at. And then finally, we have the axon terminus. The axon terminus is basically where the signal goes. So we start at the dendrite. The signal moves its way down to the terminus. And at that point, the terminus, when it receives an electrical signal, has this cue to release chemicals. And these chemicals are called neurotransmitters. So once our signal gets to the end, at that point, these vesicles open up, chemicals go out, and then they can create a new signal. More on that in just a moment. Does anybody have any question about this, the general anatomy of a neuron?
Where's its nucleus?
Its nucleus is right here. So, and this isn't the greatest drawing in the world. I'm still kind of proud of it. I drew it last night in PowerPoint. But so it's a very, very crude representation of a neuron. Although, I've seen worse. Anyway, so this is our nucleus right here. This is where all the genetic material is stored. And so there are other parts. Neurons are, pretty complicated entities. But for our purposes, basically, we just want to understand the signal goes into the dendrites, it goes down the axon, and then it works its way to this terminus. And that's where a new signal can be created through the release of chemicals. Other questions? Okay, so what makes this work? How can a signal be transferred? So it works off of two key gradients. One is an electrical gradient, and electrical gradients are things that we've looked at throughout the semester. It's basically that opposites attract rule. So an electrical gradient is created by a charge difference. And basically the idea is positive charges repel each other, like charges repel each other, opposites attract. It's the Paula Abdul rule, opposites attract. Does anybody get that reference? If you do, please type it into the chat. A number of my references are dated. I don't see anybody typing into the chat. Uh-oh. Anyway, opposites attract. So the other type of gradient we have is a chemical gradient. And this is created from a concentration difference. And it turns out this is actually kind of a function of probability and entropy. But something that we're probably familiar with is if you have some sort of gas and it's being emitted from, like if, for example, you spray perfume. We have these esters that create a smell. It starts off concentrated in a very small area and it will diffuse out. This is an example of a concentration gradient. And what it tells us is that chemicals tend to move from areas of higher concentration to areas of lower concentration. So neurons create an electrochemical gradient. They create a push on the outside of our cell and the inside of a cell. So thinking about the fact that opposites attract and like charges repel. On one side of our cell, we have something that's V concentrated with sodium. Look at all those sodiums there in our cartoon. On the other side of our cell, it's, well, not V concentrated. So which side would you say has the more positive charge based off of sodium? The outside or the inside? Which side is more positive? Which side has more of a concentration of positive charge on it? Give a thought. One moment. Okay, perfect. The concentrated side, exactly. So the concentrated side has lots and lots and lots of positive charge. All those positive charges, they're repelling each other. That's not super favorable. So if given the opportunity, some ions would want to migrate into the inside. And so this is actually an example of both an electrical gradient and a chemical gradient, an electrochemical gradient. So there's lots of positive charge on the outside of the cell. Those positive charges repel each other. If everything's intact in our cell bilayer, they're blocked. So here we have basically, you can almost think of it as being shielded. So we have the shield going, blocking the inside of the cell. But if we were to do something to remove that shield, then suddenly our sodium ions are going to whoosh, work their way in. They're going to go from more concentrated to less concentrated. They're going to go from that more positive environment to the less positive environment. And this is a byproduct of both the charge gradient and the chemical gradient. And the question was, is this similar to osmosis? It is, yes. So osmosis is the movement of water to help level out concentrations. And it's the same idea. It's just we have a membrane that instead of letting ions through, it lets water through. And water will migrate until both sides are concentrated. So basically the water will migrate to make the more concentrated side more dilute. So it'll keep going until you have equal concentration. So it's actually basically the exact same concept, except instead of ions moving, it's water moving. Excellent question. All right, so enter the idea of an action potential. An action potential is an electrical signal carried by changes in potential. So changes in voltage. And it works its way down our neuron. So what happens is we get an input at the dendrites. So we get input, input, input. And when we get enough input, it sends this signal that whoosh goes down the axon and then ends at the axon termini. Okay. And so the way that it generally works is that when we send a signal into a dendrite, so our resting potential, we said before, a neuron that's just chilling out, not really doing anything, sits at about -70 millivolts. So our neuron, before it's activated, has this resting potential. And once we send a little bit of a signal in, it starts to go up a little bit. It's not enough, though, if we just have one little signal to create a full-on response. Then we just have these signals just jolting all over the place in our bodies. But once these start to receive some more signals, so once these receive more signals, basically you can think of the dendrites as being signal collecting branches. Then we get enough of an uptick in voltage. These channels start to open up and sodium whooshes to the inside of a cell, which causes this sudden jump in the electrical transmission that goes up to about +50 millivolts. And then as time goes on, and not much time because this all takes place within about 0.02 of a second, the signal gets lower and lower and lower and lower until it eventually kind of stabilizes out at the resting signal again. So I'll go into a little bit more detail as to why this occurs. This is just kind of the generalization about how a signal is sent. The voltage suddenly spikes and then it lowers down. What is it that causes it? It's the movement of sodium, and we'll analyze that in a little bit more detail. We'll spend like the first 5 or 10 minutes or so talking about why this works on Friday. This will be a kind of our cliffhanger here. And then we'll talk a little bit about the scientific method and about drug development, research, what something has to go through to become a bona fide FDA-approved drug. And yeah, I'm excited. So I hope that you all have a wonderful rest of the day. Thank you, as always, for your time. Thank you again for your continued awesome efforts. You know, the switch to everything online is certainly not easy. I can't imagine what it'd be like to have, 5, 6, 7 classes, and then suddenly everything is turned upside down. But you should be very proud with how resiliently you've handled it, how adaptive you've been, and just how generally awesome. So thank you for your continued hard work and for just being a great, nice group. And hope that you have a wonderful rest of the day. I'll see you on Friday.